Right now, there are living human beings with genetically modified pig organs beating inside their chests. We have officially crossed the species barrier. This chart shows the widening gap between patients on transplant waiting lists and the available supply of human donors. It represents a mathematical tragedy. Thousands die every year because available human organs remain largely stagnant while demand climbs. Pigs have long been considered an ideal anatomical match to solve this. Their organs are the right size and function similarly to ours. But for decades, doctors hit a violent biological wall called hyperacute rejection. If you place a wild-type, unmodified pig organ into a primate, the host's immune system destroys the tissue within minutes. Closing that gap was never going to happen by waiting for more human donors. To solve the shortage, scientists realized they had to hack the underlying biology of the donor animal itself. The tool that made this possible is CRISPR-Cas9. It acts like molecular scissors, allowing scientists to alter DNA with enough precision to create multi-transgenic pigs. The engineering requires a two-part genetic hack. First, they knock out the pig genes that produce gal antigens, which trigger hyperacute rejection. Second, they insert human protective genes that help the organ regulate blood clotting and immune responses within a human host. In recent preclinical trials, these specific modifications allowed pig kidneys to sustain life in non-human primates for over a year and pig hearts for up to 9 months. The bioengineering is a success in the lab, but moving these organs from primates to human patients moves the challenge away from the genetic code and toward the complex legal and medical frameworks that govern human health. These groundbreaking surgeries are happening right now at major medical centers without the traditional safety net of official clinical trials. To bypass the strict restrictions on living human trials, researchers first turned to the Decedent model. They transplant genetically modified pig kidneys into brain-dead human patients to study organ function and immune responses without risking a living person's life. When it comes to living patients, the path is even narrower. The FDA has authorized a handful of experimental transplants under its expanded access pathway, often called compassionate use. This is a special allowance granted only when a patient has a severe, life-threatening illness and no other survival options. These compassionate use cases are providing real-world proof that pig organs can sustain human life for months. If you are finding this intersection of biology and policy fascinating, take a second to like this video and subscribe to the channel. While these regulatory allowances are keeping desperate people alive today, they are also revealing a new layer of immune rejection. Putting these organs into medically complex humans has exposed biological variables that tightly controlled lab environments could not predict. The immune system divides into two branches: the adaptive T cells and the innate macrophages. Even with CRISPR edits, the body fights back. Standard drugs suppress the adaptive response, but the innate system remains active and continues to attack the graft. The physical consequence of this innate attack is microvascular thrombosis. The ongoing inflammation causes blood clots to form inside the tiny vessels of the organ, eventually blocking blood flow and destroying the tissue from the inside out. There is also an invisible threat: porcine endogenous retroviruses, or PERVs. These are retroviruses embedded directly in the pig's own DNA. If these viruses leap to human cells, they could trigger a cross-species zoonotic infection. Because of that zoonotic risk, a failed xenotransplantation is not isolated to the individual patient. If an animal-to-human transplant introduces a novel pathogen into the human population, it becomes a biological threat to public health. Single one-off transplants under compassionate use protocols provide vital snapshots of data, but they cannot provide the statistical certainty required to make xenotransplantation a standard medical procedure. In response, federal regulators are stepping in. The FDA's 2026 guidance agenda includes draft frameworks specifically aimed at establishing official, highly regulated xenotransplantation clinical trials for solid organs. These new guidelines mandate strict operational protocols. To minimize infection risks, donor animals must be bred and maintained in highly controlled, biosecure facilities. Furthermore, recipients will require lifelong monitoring and biosurveillance to track any potential pathogen transmission. Regulators are forcing xenotransplantation out of the experimental boundary of compassionate use and into a standardized, rigorously controlled clinical system. This transition exposes the complexity of informed consent. If a patient is facing certain death with no other therapeutic alternatives, the line between a voluntary choice and a desperate survival instinct becomes incredibly thin. There is also the danger of xenotourism, the risk that desperate patients will travel to countries with weaker biosafety oversight to receive experimental animal organs if domestic rules delay their access. Beyond medical and legal risks, the practice raises questions about the status of the animals themselves. We have to confront the implications of breeding genetically altered, sentient animals solely to harvest their parts for human use. Recent clinical cases have demonstrated that a genetically edited pig organ can function within a human body. The biological feasibility has been shown. But as we move forward, we have to decide if our laws and ethics are prepared for the consequences of blurring the line between species.